right, today we're going to talk to you guys about signaling pathways between cancer stem cells within triple-negative breast cancer and treatment options.
Right, so today we're going to be talking about what breast cancer is, specifically what triple-negative breast cancer is, and what are the cancer stem cells that are within breast cancer, and specifically the breast cancer subtype stem cells that make breast cancers harder to treat. We're also going to talk about the epithelial to mesenchymal transition and the epithelial to periocyte transition, which play a role in inducing stemness into cancer cells. And then we're going to identify different signaling pathways being targeted in order to treat cancer cells and share research of a mechanoresponsive vector created in order to deliver treatment specifically to cancer stem cells.
All right, so breast cancer is the second most common cancer diagnosed around the world, and it most commonly occurs in women. The cancer cells occur in the lobules or ducts that connect lobules to the mammary papilla. So if you look here in this pink area, these are the lobules, and these are the ducts that connect to the nipple, which is also known as the mammary papilla, and this is where the cancer tumors will reside. There are many subtypes of breast cancer that are based upon genetic and epigenetic variables due to cancer cells, and these four subtypes are luminal A, luminal B, HER2-enriched, and triple-negative basal-like, which is the one that we're going to be focusing on today. These subtypes are all based upon whether or not they express these hormone receptors or human epidermal growth receptors, and allow them to be classified into these different subtypes. Triple-negative breast cancer is commonly diagnosed in women that is less than 40 years old, and 15 to 20% of women who are diagnosed with breast cancer have triple-negative breast cancer. TNBC is extremely hard to treat due to the fact that they're lacking both of the hormone receptors and human epidermal growth receptors 2 that we looked at on the last slide. Due to the lack of these receptors, they are not able to be treated with common hormone therapies that are being used to treat the other subtypes of cancer, leading many problems in trying to alleviate this disease within individuals. It's also super difficult to treat due to the presence of cancer stem cells and breast cancer subtype stem cells. As a result, they cause regrowth of tumor after conventional cytotoxic treatments such as radiation and chemotherapy. And often they lead to metastasis after treatment, which is just the migration of cancer cells throughout the body from the original location. So cancer stem cells have the ability to self-renew and proliferate under non-adherent and non-differentiating conditions, which basically means that they kind of play by their own rules, and they regulate the signaling pathways in different ways to adhere to their viability and to promote the cancer stem cells within individuals. And cancer stem cells are most commonly formed by the epithelial to mesenchymal transition, which is most often followed by the epithelial to parasite transition that we'll discuss on the next slide. And breast cancer stem cells are a derivative of cancer stem cells that are seen within breast tissue, and they often lead to a worse prognosis within patients who have triple-negative breast cancer. And as well, they also cause tumorigenesis after treatment. So the epithelial to mesenchymal transition and epithelial to parasite transition are a major cause for stemness that is seen in cancer cells and is a main reason for regrowth of tumor cells after treatment. So here you can see the normal epithelial cancer cells, and as they undergo the epithelial to mesenchymal transition, these epithelial cells now have a mesenchymal phenotype, which often leads to an increased migration and aggression of cancer cells. After this transition, they will undergo the epithelial to parasite transition, which changes the mesenchymal phenotype and transforms it into a parasite phenotype, which allows these cells to undergo PGF signaling, which essentially allows these cells to associate with blood vessels, and it brings blood to carrier cells that will then bring it to the tumor cells to help cause regrowth of tumor. Now we're going to talk to you guys about the specific targeted pathways and treatments that researchers have been looking into in the past couple of years. Mainly the inhibition to signaling pathways has posed new methods of treatment for triple-negative breast cancer, and the majority of these research has focused on these specific pathways: the cyclin-dependent kinase 2 and 4 within the SMAD3 pathway, the NF-κB signaling pathway, and the WNT beta-catenin signaling pathway. Also, we're going to discuss the mechanoresponsive treatment within mesenchymal stem cells that will deliver treatments directly to cancer stem cells in order to induce apoptosis. So the pathway of the cyclin-dependent kinases within the SMAD3 pathway have two different options that it can go. First, it will go through the canonical pathway, which increases tumor suppression and decreases EMT activity, and this is most commonly seen in individuals who are healthy and don't have any underlying health conditions. However, in patients who have triple-negative breast cancer, the SMAD3 pathway will resort to the non-canonical pathway, which has high expressions of cyclin DE. These high expressions will cause an increased activity of cyclin-dependent kinases 2 and 4, which then leads these kinases to phosphorylate the transforming growth beta factor. And when this happens, it can lead to oncogenesis. The phosphorylation of transforming growth factors beta also catalyzes the interaction between SMAD3 and PIN1, where PIN1 is an isomerase that is overly expressed in patients with breast cancer, and this also leads to oncogenesis as well as migration of cancer cells. If we go back up here to cyclin-dependent kinases 2 and 4, it also causes a dysregulation of cyclin D1, which also results in increased oncogenesis and migration of cancer cells, which in the end promotes the ability of breast cancer stem cells to remain alive. All right, so they have looked into the inhibition of the cyclin-dependent kinases through two different research studies that allows the SMAD3 pathway to resort away from the non-canonical pathway that promotes breast cancer stem cells. In the first study, they used cyclin-dependent kinases 2 and 4 inhibitors, which prevented the phosphorylation of SMAD3, thus resulting in restoring the tumor suppressive signaling and decreasing the size of tumors. In the second study, they used a cyclin-dependent kinase 2 inhibitor and a PIN1 inhibitor and used mice as their study organism. In this study, they used SMAD3 phosphomutant T179 as a vector, which has the ability to replace a threonine with a valine at the T179 site of cyclin-dependent kinase 2, which causes cyclin-dependent kinase 2 to not be phosphorylized. This is then followed by the inhibition of PIN1 by a PIN1 specific small interfering RNA, which seizes SMAD3 PIN interaction, which results in triple negative breast cancer to be less aggressive in limiting migration signaling pathways. This study proved to be successful in mice.
So another pathway being targeted within cancer stem cells is the NFKB signaling pathway, or the nuclear factor capital light enhancer of activated B cells. It's a long name. But the end result is that it produces transcription factors P52 and RELB, which are down here, which go into the nucleus and promote the transcription of cell survival proteins, which then promote exponential cell growth and kind of induce cancerous properties into cells. So it starts when a ligand binds the tumor necrosis factor receptors on the cell membrane, which initiates a cascade of phosphorylation, starting with enzyme NAK and then to IKK alpha, which then phosphorylates the complex containing P100, a protein, and RELB, another protein. So then this complex gets degraded by a proteasome, producing P52 and RELB, which can then go into the nucleus and promote transcription. However, this pathway is being researched into finding ways to inhibit it, to stop it from inducing cell growth, and find ways to induce apoptosis in cancer cells. So research has found that curcumin, an organic compound extracted from turmeric, it's actually what gives turmeric its yellow color, is able to decrease the expression of acyl receptor tyrosine kinase, which is a upstream regulator of the NFKB signaling pathway. And this acyl receptor tyrosine kinase also has the ability to induce rapid cell proliferation. So curcumin prevents the activation of this upstream regulator of the NFKB pathway. Therefore, it prevents the synthesis of transcription factors being made through the NFKB signaling. And it also prevents acyl receptor tyrosine kinase from inducing rapid cell proliferation into cancer cells and making the cells more likely to undergo apoptosis. So another pathway being researched is the WNT/beta-catenin signaling pathway. And this one is prominent in 50% of breast cancers, specifically in triple negative breast cancer. And it typically leads to a worse prognosis. It's also what allows cancer stem cells to maintain their stemness by promoting the transcription of genes that elongate telomeres, such as the TERT gene. So beta-catenin, when it binds the promoter of the TERT gene during transcription, it will increase the expression of the TERT gene, which functions to elongate telomeres in cancer stem cells, promoting their viability. So this pathway begins when WNT ligands bind the LRP and FZD protein receptors on the cell membrane, initiating a cascade of phosphorylation that ultimately ends with the enzyme glycogen synthase kinase 3 beta becoming deactivated when it gets phosphorylated. So you can see here that it is no longer able to bind or process the extra unphosphorylated beta-catenin, shown here in blue. So this causes a buildup of the beta-catenin, which then gets translocated into the nucleus and is able to activate the transcription of cell survival genes, such as the TERT gene and others. So this pathway's research into this pathway has research into inhibiting this pathway and has found that it's been able to induce apoptosis in the cancer stem cells that exhibit this pathway. One method of inhibiting this pathway is using proteagestin, which inhibits the substrate phosphorylation of LRP6 and DVL2, which are just intermediate substrates in this phosphorylation cascade. And proteagestin also activates the enzyme glycogen synthase kinase 3 beta. And once activated, this will process all the buildup of excess B-catenin and prevent it from going into the nucleus to activate transcription. So proteagestin has also been shown to deregulate just another gene that aids in cell survival, BRC5, or the cell survivin gene, that if translated, it would create a protein that is anti-apoptosis. So the whole goal of targeting this pathway is to prevent the transcription of cell survival genes and make the cells more likely to undergo apoptosis. So another method of treatment being researched is a way to specifically target the cancer stem cells within the tumor microenvironment instead of just inhibiting pathways that they have. So researchers have used mesenchymal stem cells for their ability to sense their environment and settle in stiffer environments. So one way they're able to know when to settle is the presence of the YES-associated protein, or YAP, which settles or goes into the nucleus and becomes activated as a transcription factor in environments more dense than 10 kilopascals. But in environments less dense than 10 kilopascals, it goes into the cytosol and becomes degraded. So the presence of YAP allows mesenchymal stem cells to sense their environment and be able to translocate or be able to settle in the tumor microenvironment. So these researchers used cytosine deaminase and in mesenchymal stem cells, and then when inserted into the test subjects, these mesenchymal stem cells settled in the tumor microenvironment, shown here, and released cytosine deaminase into the cancer stem cells that had already been treated with 5-fluorouracil. So the presence of both of these drugs caused apoptosis of the cancer stem cells within the tumor microenvironment. So this is one treatment that provides hope and light to finding a more reliable treatment for breast cancer.
Due to the fact that cancer stem cells are present in triple negative breast cancer and the fact that it lacks hormone receptors and human epidermal growth receptors, triple negative breast cancer has been extremely difficult to treat in the past with not many viable options. Although current research shows hope that a viable option will soon undergo clinical trials and be approved for patient use, but for that to happen, much more research must be done in order to discover a cure that will alleviate all individuals who are affected by this disease. Hopefully, in the up and coming future, there will be a viable treatment for these individuals. Thank you guys.